The much-awaited webinar lecture will be delivered by none other than Dr. K.V. Radhakrishnan, sir, Senior Principal Scientist of CSIR-NIIST, Thiruvananthapuram. Dr. K.V. Radhakrishnan obtained his PhD degree in Synthetic Organic Chemistry from the University of Kerala in 1998 at CSIR-NIIST, Thiruvananthapuram. Subsequently, he held postdoctoral positions at Tohoku University in Japan, at the Molecumetrics Institute and NGP Research Institute in the United States of America. He joined CSIR-NIIST as a scientist in the Chemical Sciences and Technology Division in May 2002. His research interests include synthetic carbohydrate chemistry, transition metal catalyzed organic transformations towards pharmaceutically important molecules, organic phytochemistry and photochemistry with a special focus on bioprospecting of medicinal plants from the Western Ghats. He has guided about 30 PhD students and is currently guiding 6. He is here with us today to deliver the talk titled Nature's Inspiring Revelations for Future Technological Developments. On behalf of the Kerala Academy of Sciences, I extend my welcome to Dr. K.V. Radhakrishnan. Sir, you can now take over the session.
Okay, thank you. Can you unshare this already shared thing? May I share now?
Sure, sir.
So, okay. Let me try. Can you see the slide?
Not yet, sir. Yeah, it's coming.
Is it okay?
Yeah, it's coming.
Okay,
You can put it full screen now.
So,
Yeah.
okay.
Nice, nice.
thank you. Yeah.
How is it now?
Yeah, it's good. You
Fine.
can start now, sir. Yeah.
It is in full slide mode?
Yes.
Yes.
Because today morning the network has a problem. That is why I couldn't join. So thank you very much, Ramesh, and please stop me if I'm taking more time. So I have adjusted my talk to around 20-25 minutes maybe. So first of all, let me thank KS, especially Professor J.M. Nair and Dr. Ramesh for inviting me for this talk on phytochemistry. So when I got invitation, I was asking Dr. Ramesh, what should I talk? Should I talk about the isolation and characterization or the pharmacology of isolated molecules which we are doing? So at the outset, let me tell you that I am not trained as a phytochemist, but it is the passion for botany which has led to this area, and it is the passion for botany which is driving me in this area. So now can you see the slide? Next slide. I'm in next slide.
Yes, sir. Yes.
Okay. So at the outset, I would like to dedicate this talk to Dr. Vijay Nair, who is celebrating his 80th birthday tomorrow for his celebration with years of harmony with organic chemistry. So whatever I'm going to talk to you about is a multidisciplinary activity at chemistry biology interface, and it is not a single person work. It is a team work where many persons have contributed, starting from MSSRF, JNTBGRI, RGCB, CDRI, and several people are involved in this one. And that is why there is a lot of pharmacology is also involved, but I will not be presenting those aspects. So when it comes to the richness of Indian biodiversity, we are so rich because we have this Himalaya and the northeastern and western Ghats. We are so rich. And when you look at this western Ghats, which is one of the 8 hottest hotspot of biodiversity, we are so fortunate to be part of this area, which starts from Nagargovil to the Gujarat, which is a rich source of a number of flora and fauna, which all of you know. This is an exciting thing, and this is, and I feel that this is the treasure of India, which can aid in the economic progress of India. So far what we did is we have done the chemoprofiling of different families of plants, around 45 families and 125 species of plants used in traditional medicine. And it was actually a war-footing exercise for me, especially because in the last 6 years, we were trying to learn phytochemistry. And we did it in a way, in a time-bound manner also, utilizing all the instruments which are available with us. I have to say that because to do phytochemistry, it is time-consuming, but to do it faster, you need the support of instrument, especially for the isolation and characterization. So to start with, I would like to start with our ginger family, Zingiberaceae family. So some of the main plants which we have investigated in deep is Zingiberaceae family, wherein two genus from Zingiber genus, Zingiber cerumbet, Zingiber nimeni, and another one is the Hedychium flavescens. Curcuma, several curcuma species have been investigated in the lab. I will be talking just about this plants, and my senior and colleague Dr. Mangalam Nair has also worked on a number of other plants in this species, Curcuma originosa, Curcuma rectagenda, etc. So if you look at the compounds isolated from this family, Zingiberaceae family, you can see there are humulines, cerumbones and cerumbols, and then comes to different types of sesquiterpenoids like, let me put that point, like germacron, furanoylamine, and all those things. So as an organic chemist, I get excited with the structures because this structural characterization is challenging and also at the same time exciting also. So a different type of molecules, and all I want to mention here that all these molecules are isolated through a cold destruction process, because all these molecules in Zingiberaceae especially, it undergoes other side reactions if you heat it. Even for Clevenger apparatus, when you heat it, these compounds gets converted to many other molecules, especially some azulines. It undergoes elimination reactions and so on. So coming to another family which we have worked on a lot is Dipterocarpaceae family. Dipterocarpaceae family is mostly trees, and some of the plants which we worked on in Dipterocarpaceae is Vateria indica, which is commonly known as Vella pine, and Hopia ponga or Kambagam, and then Vatica chinensis or Adacia pine, and then there is another one called Hopia parviflora. So all these plants are widely utilized in folklore remedies of Kerala, mainly, especially the resin coming out of it is used as an additive in a number of folklore remedies in Kerala. So you look at it, the compounds. Sorry for showing all these structures. For some of you maybe interesting. But in Dipterocarpaceae family, if you look at the main compound in Dipterocarpaceae family is resveratrol. That's the monomer. So when the plant grows, this resveratrol undergoes several biochemical process. It undergoes dimerization, forming viniferin, or if it is in Vatica chinensis, it undergoes gets into ambulopsin and different types. So the resveratrol undergoes different type of biochemical processes, getting transformed into viniferin, ambulopsin like dimers, and 3 molecules of resveratrol can get together in different forms, of course, in a trimer form, and 4 of them can form tetramers in the form of hopiaphenol, vaticanol, etc. And also it can form tetramers also. So you can see these complex structures. Though it is complex, the characterization is really exciting and challenging too. So coming to, so you might have seen in these pictures, I have put minus in front of all names, like minus viniferin, minus ambulopsin, minus hopiaphenol. So I want to tell you that what you are getting in all these plants in Dipterocarpaceae family is minus quantity. it's a optical rotation. So let us come to Whiteaceae family. Whiteaceae family, we mainly worked on two plants, that is Ambelosis indica and Cissus quadrangularis, which is Cissus quadrangularis is widely used in Ayurveda and it's a well-studied plant. So we have worked on plants which are already known in literature also, because that is mainly for utilizing the phytochemicals in it for scientific validation of Ayurveda drugs and herbal formulation, which we usually do as a consultancy for Ayurveda companies. So when you come to Ambelosis indica and Cissus quadrangularis, you can see this over here also. In Whiteaceae family, you may know wine is a rich source of resveratrol, and resveratrol is the monomer, and you can see it can form the dimer like viniferin and tetramer. It has hopiaphenols and trimers, etc. And you can see these are all the mirror images of compounds which are found in Dictyrocarpaceae family. These are all plus. So Whiteaceae family is having plus hopiaphenol, whereas the other one, the Dictyrocarpaceae is having its mirror images. So it was really exciting. I will come to the characterization part at the end of the talk. We have different types of flavonoids also, quercetin, isoquercetin, etc. From this plant. And another plant of interest is a Samarobaceae family, and wherein we studied several plants. I am just talking about two plants here. Cosia indica, in Malayalam it is known as Carignota, and Ailanthus trifisa or Perimera. So both are used in folklore remedies, and mainly it is used as anti-inflammatory, so especially in rheumatic complaints. And look at these molecules. So when you take Cosia indica, these are the molecules like cosinoids. This structure is a special structure. The lactone structure is known as cosinoids, and you can see some are there in A, B, C, etc. And we were able to isolate a number of novel molecules from this plant. And you have to note a point here that depending on the geography and the soil type, the molecules, phytochemical combination also varies. Sometimes you get different type of phytochemicals when you collect it from a different geographical location. So when it comes to Ailanthus trifisa, we are still working on it. It has several novel molecules. Only two of them we were able to characterize, because the characterization is really time consuming for us now. Ocotilone and similar molecules like this triterpenoids. And we have also worked on Musaceae family, the Musa bulbiciana, the seeded banana family with Dr. Sabu of Calicut University. And in the seed is having the major compound as epiferol and a number of other compounds which we were not able to characterize so far. And the rhizome of the banana, the rhizome is having this type of compound, especially when the rhizome is infected with many different insects are infecting, it is having this interesting fluorescent compounds. Very interesting, but very little we get from the plant. And there is another family called Celastraceae, which we have interest mainly because of the our activity in the area of CNS disorders, as memory enhancing and also in skin inflammation. We have studied a plant called Celastrus paniculatus along with the Nordostachys jadamansi and the Sarcostem acidum, which is Somalada. That is what we are studying right now. And you can see we have isolated a number of molecules, and the molecular structure, as you see, it is agarofurane. Agarofurane is nothing but two cyclohexane ring and a furan ring fused together. It is called agarofurane. We have isolated several molecules, 3 novel molecules along with 3 novel and 9 non-molecules. So almost all of them, the structure is similar, only the stereochemistry in each carbon is different. So coming to Moraceae family, Moraceae, when it comes to Moraceae, the Kerala people really get excited because it is a family of jackfruit. The jackfruit is Artocarpus heterophilus, but Artocarpus, there are many other species. Artocarpus heterophilus is a well-studied family, but there are many other species found in Kerala Western Ghats. Artocarpus camansi, Altilis, the breadfruit. Camansi is not known as bread nut fruit. Artocarpus lacucha, and also Artocarpus hirsutus or Anili or Aini. And in all these families, one of the main compound in, one second. One of the main compound in all these Artocarpus genus is oxyresveratrol. So oxyresveratrol, what is the use of oxyresveratrol? Let me tell you. This is oxyresveratrol. And if you look at the face whitening cream and skin cream application, you can see 0.002% of oxyresveratrol. It's one of the main component in a number of cosmetic creams. And this is being exported from Kerala to many places also. So not only that, this family is having a number of rare and exciting molecules, wonderful molecules like Kudreflavone, Artocarpin, etc. And you can see this is having, it is like our broom. You can see it is like our broom. It is a highly antioxidant, and the broom, this this type of structures, it is like like a scavenger actually. So this type of appendages in the molecule helps in the radical scavenging. It is an antioxidant, so antioxidant, and these wonderful molecules, artonin, artonin A, artonin B, and etc. And I'm not going too deep into these structures. And another wonderful family, especially in times of COVID, is Menispermaceae family or moonseed family, because it has heart-shaped leaves and moonseed. So over here, where what we have worked on is Cocinium fenestratum, which is otherwise known as wood turmeric. It's an endangered species. And Tinospora cordifolia is a well-explored plant. And then Cyclia peltata, Tinospora crispa, which is a duplicate of Tinospora cordifolia. And those who doesn't know the morphology of Tinospora cordifolia always go for crispa, getting into liver problems, because most of the compounds in Tinospora crispa is toxic, hepatotoxic. And another one which is used in folklore medicine, Stylacora acuminata or Pollicandida. And all these plants and many Menispermaceae species, along with the steroidal compounds, one of the main compounds in this family is alkaloids. And if you look at the alkaloids in Cocinium fenestratum, berberin. Berberin gives that yellow color and so nice molecule, and you get in grams from this plant. From 1 kilogram of the dry weight, you can get 3-4 grams of berberin, oxyberberin, etc. And when it comes to Cyclia peltata, you can see Cyclia peltata and Stylacora acuminata. These are plants which gives cyclic alkaloids. It is called bis-benzoquinolone alkaloids, cyclianin and fiandin. You can see fiandin. Fiandin is the mirror image of tetrandrin. Tetrandrin is one of the alkaloid from this family, which is undergoing second phase of clinical trials in China against COVID-19. And then coming to Myristicaceae family, Myristica fragrans, Myristica malabarica, Myristica bedomi, and really Kerala, Western Ghats is having a rich source of all this one. And when it comes to Myristica fragrans, it is cultivated commercially here. But at the same time, there are a number of other wild species called Malabarica, Bedomi, Fatwa, and most recently the Myristica tropicata. This is in collaboration with TVPM, right? And when you look at the structural aspect, fragrance is having mainly lignans, which has antibacterial, antimicrobial, etc. Whereas all wild varieties of Myristica species is having acylphenols. And some of these acylphenols are so toxic also and allergic to many people, just like cherry marum. And more than that, cherry marum allergy, I mean, you can get rid very easily within a week. But the allergy coming from this wild Myristicaceae family seed and some of the compounds are so bad actually. You have to have a PPE kit to work on these plants. And another family of interest to us is Delineaceae family, where we worked on Delinea indica and Delinea pentagina, and and we worked on 2 different geographical locations from Kerala and also from Assam, and became a marker of this species, Dilene indica and pentagynase betulinic acid. When we collected it from Assam, we had some other compounds also, which is very good actually, quite jabic acid and palmaromycin. And another family of interest was Phyllanthacea family. It doesn't have much exciting molecules, so but Brydelia retus and Brydelia stipularis, it has a lot of gallic acids and ellagic acid, etc. And another interesting family which has interest both in folklore medicines and in Ayurveda is Cipitasa basifera, otherwise known as Kaipanaranyi in Malayalam. It is used only in folklore medicines as against snake poison. And Aphanamaxis polystachys or Chemmeram, which is it's called the Rohituka savam in Ayurveda, mainly used in spleen disorder. It is prepared from, it is having this Aphanamaxis polystachya. So the Aphanamaxis polystachya is being introduced to me by Dr. Asha Leda Madam of Kerala University Botany Department. And both these families, if you look at the Cipitasa family, is a rich source of leminoids and triterpenoids, whereas the Aphanamaxis polystachya is having a number of compounds. Still, we are isolating a number of compounds from it. Isolated, all the compounds are isolated, but the characterization is going on. And another one, the Bougainvillea family, Nyctanginaceae family, we worked on the phorochrome plant with white color, the rhizomes of it, and we were able to isolate several rotenoids. This particular skeleton is known as rotenoids. And Fabaceae also, the Fabaceae we worked on well-known plants, 2 well-known plants, mainly to aid in our consultancy projects with Ayurveda and also collaboration with Ayurveda Medical College, wherein we isolated several flavonoids and carbohydrate-appended flavonoids as well. And another family which we were working on is Astraceae family. I would like to tell you about 2 of them where we have just completed the isolation characterization, Vernonia anthelmintica and Vernonia cinerea. Both are used in Ayurveda, and Vernonia cinerea is one of the flower used in Dashapushpa. So Vernonia anthelmintica, the seed is used in Ayurveda mainly, and as all of you know, Vernonia cinerea or Puangurunella is also well known to all of you. So I don't want to give it. So look at these compounds. So when it comes to the compounds, I get excited because the characterization is challenging. So so we get into the characterization at last to know that it is already known, but doesn't matter. But the biology, pharmacology of these molecules are not known. So but not in the same species. So that's all about the isolation part. And Dr. Ramesh told me I should give a note on the isolation characterization and also. So these are some of the main families we worked on. In fact, we worked on a number of other plants. I'll not be able to talk about all of it. It will be boring with all these organic chemistry structures. So let us come to the plant collection, extraction, and isolation. So plant collection, extraction, and isolation. When we are going together and as a team, it is really interesting. Otherwise, it is not so interesting. So you have especially the bark collection, and then you cut it and dry it and powder it. So mainly, we usually go for cold extraction, just to make sure that we are not losing anything on heating. So the cold extraction, after powdering, we put it into bigger RBs, or we just soak it and then filter it. So once we soak it and filter it and then evaporate, and the residue, we just run the first thing to do is the residue is used for running a short column, short and big wide column, where we are just filtering it and storing it in different boiling tubes, a number of boiling tubes. where and then because the students or myself is not having patience to go for long columns which takes 2 months or 3 months, and students will not join them. So for PhD or any work. So what we do is we cool it down. We use the refrigerators which is available, which you can see in supermarkets, and we adjust the temperature, and then we do the precipitation, or sometimes the viscous things settle down, and then we go for the crystallization. Only when it is not possible to precipitate or use it, or you cannot settle it, then we go for column chromatography techniques, where we use different types of column materials like silica, alumina, sephadex, dianon, depending on the type of compound. And sometimes you use proprietary TLC also. And one of the main help which we get is from flash column. We had several flash columns in the institute which was not in functional and all. And what we did is, when we started it, we repaired all of it, almost 4 of them. And when all these 4 are running, we can, once you complete the extraction, and you can finish the isolation characterization in just 4 months time. And in addition to that, we have a recycling 2 recycling HPLCs, which is from the JHPLC of Japan, tech science company, and then proprietary HPLCs, analytical HPLCs. And HPLCs are used only when it is really required, otherwise, because it takes some time for the purification. So the characterization, the first thing as for a phytochemist to do is the melting point to find out the purity. And then if you want to find out which are the functional groups, you have to go for IR, and then chromophore identification through UV absorption. And the next most important thing is high resolution mass, the molecular mass identification. So through molecular mass identification, you can, come to the idea of some of the around 10-20 molecules having the similar molecular mass from the same family. Elemental analysis also, NMR, and optical rotation to find out the relative configuration. NMR for structure elucidation, optical rotation for relative configuration of all the molecules, and then single crystal XRD if the compound is crystalline, absolute configuration, and then circular dichroism. So the NMR high resolution mass is very important because it gives the molecular ion mass of, and also it gives the fragmentation. When it is ionized, it gets fragmented. You can get to you can look at the fragment ions to see which is which, and the fragments give you an idea what is the actual compound. So by analyzing the fragments, you will get an idea about what type of compound it is and where it is connected, where it is getting cleaved, because the cleaving takes place at the weaker point. So, for example, an example is that hexamer of resveratrol from Diteroccarpaceae family. You can see hexamer mass and then tetramer mass and dimer mass over here, which I have noted here. So this is a systematic process, very exciting, and it can be done by anybody, and it is all done by my students in the laboratory, especially the master's students also. And when it comes to the structure elucidation, we had to utilize several types of NMR techniques. What is a normal proton NMR, carbon NMR, and then proton correlation to see which proton is neighboring to which other proton, neighboring proton-proton interaction studies. And then you have to go for the 2D NMR techniques like HMQC, HSQC, etc., which gives which carbon is connected, which proton is connected to which carbon. Okay. And another one is HMBC technique, which gives an idea about the neighbors. It is not the direct connection. You will get an idea about who is in your neighbor and that neighbor. So who is in that line? So for example, you can get 2-bond correlation, 3-bond correlation, up to 4-bond correlation. So that is the idea of HMBC technique. And to find out the stereochemistry, we have to go for the noisy experiments, and also to find out the hybridization of carbons and how many not only how many protons are attached to each carbon, you have to go for depth experiment. I will go one by one. So this is one of the novel molecule, and this is how it looks like. So if you are not able to do a systematic study, it is just like getting an analysis result, like image from a medical examination. But we had to, it's not difficult. So look at the carbon NMR of the same compound. So you can see carbon NMR, and by doing several types of depth experiments, you will get how many carbons are having only one proton attached, how many carbons are having 2 protons attached, and how many carbons are having 3 protons attached, and how many are having no protons attached. So this gives an idea. So that means primary carbon, secondary carbon, tertiary carbon. So all this idea you get from depth experiments. And then comes which proton is related to another protons. This is coming from homocosy, proton-proton correlation. So where we will get an, this is a 2D NMR experiment. You can see, and the connection is ascertained through the 2D NMR experiment. You can see the squares. Squares indicate the connection between this. So we have to extrapolate it to the peak, which will give you an idea about which proton is connected to which proton. Okay, proton interaction. And then you have to go for the HMQC, gives which proton is connected to which carbon. This is also a 2D NMR experiment. So you have on one axis is the proton NMR and another axis is the carbon NMR. And so you will get which carbon you can, from the experiment, you can see which carbon is connected to which proton. And heteronuclear multiple bond correlation. This so you don't know you cannot understand anything about your immediate neighbor, but you will get an idea about your neighbor's immediate neighbor or his immediate neighbor. So that is what is the use of HMBC experiment shows. It is not the direct interaction, but you can get the second home in the line and the third to your home and the fourth to your home. So that's it. And then to find out the spatial structure of the molecule, we have to go for nuclear Overhauser effect or NOE. So once you get, if you are fortunate to get a crystal, you can get a crystalline single crystal X-ray, which will give the absolute configuration of the molecule. If it is not having any other, hint on what the molecule is, then you have to go for some more techniques. For example, this is a molecule called a Udesmain skeleton. We don't, when we got it through a transformation, we were not having any idea on what type of molecule it is. So what we do is, we do all types of NMR experiments to see who is your neighbor, with whom you are connected, what type of proton, what colored shirt you are wearing, and what type of shirt your neighbor is wearing, and then the other neighbor is wearing. So then you get, and you had to collect all these stories together, and you had to correlate. So he is connected to him, that person. So he is then connected to some other person. So then you have to connect all this information together to get an idea about what the structure is. And once you get that structure, after getting, then you have to have a absolute configuration coming, the stereochemistry is coming. So what you do is, so you draw a plausible structure, and then you will have an idea which proton is connected to which carbon and all those things. And then you do is, you shoot the next neighbor, okay? Then, so neighboring protons, shooting in the sense, I mean in NMR, just for your understanding. So then the connection between the one proton and neighbor is breaking. You irradiate it, and then irradiated proton will come down, and you can see this through irradiation, and then you can ascertain which type of what type of spatial structure it is having. So, and when you are having mirror images, I was mentioning about. I will finish in 2 minutes. when you are having mirror images, for example, plus hopiophenol and minus hopium, both are same structures, same proton NMR, same carbon NMR, everything okay, but only a slight difference in 2D spatial interaction. So we then what we did is we crystallized both of it. We were able to get plus hopiophenol crystal and also minus hopiophenol. Crystalline structure, it is mirror image. And then to finally find out the absolute configuration, you had to go for the circular dichroism. And this is same with the characterization of molecules with only which are not having any proton. How will you find out? If you are having only oxygens attached, and if it is a spiral carbon, you have to resort to some other techniques to characterize. So GCS-MS is also important, especially when you are working on essential oils and triglycerides. So when we get triglyceride, you can do the saponification and methylation to fatty esters, and then esters can be estimated easily through using GCS, but we are not doing much in this area. So, and another, as an organic chemist, when we get abundant phytomolecules, what we do is we take it further for characterization. What we are thinking is we take these molecules, abundant molecules, and convert it into different types of natural products through organic transformations, very easy transformations. That is what is shown here. Cerumbone, which is a very important molecule in where cerumbate is transformed to several molecules. And also photo-induced transformation. As you can see, most of these molecules are formed through not only through biochemical processes, and also it is coming through photo transformations also. So what we did is, for example, if you take cerumbone and just put it in sunlight with then acidic catalyst, you get another natural product, core. And the same cerumbone you convert a little bit and convert in epoxide and put it in sunlight, you get another natural products. So the reason for because it is high time to look at on short-term crops and abundant molecules in short-term crops for making synthons for pharmaceutical industry. We cannot. I don't know how long we can depend on the petroleum resources for getting several of these synthons for pharmaceutical industry, intermediates for pharmaceutical industry. In fact, there are a number of molecules which are abundant from short-term crops like Zingiberaceae family and and herbs, which can be easily converted through photochemistry into different types of very important molecules. So, so to be frank, it was a very nice journey, and really enjoyed botany and also the biology, pharmacology. And in fact, students want publications. It is all published in different journals like ACS Pharmacology, Journal of Natural Products, etc. And what we learned in this last 6 years, to be frank, is the expertise in characterization of phytochemicals and microbial natural products. And when we are able to generate a rich repository, which is being utilized for undertaking consultancy projects from different companies like AVT and many other companies. And in the process, we found out that we need to have our own facility for biological screening. And so for in vitro screening, we have, with the help of my colleague, Dr. Kaustubh Kumar Maiti, we were able to establish a very good screening facility, biological in vitro facility. And also we have recently initiated a herbal product formulation facility. And in this area, this is the outcome for translation to patents filed for 2 molecules, which shows a very good synergistic activity against MDR strains of Staph aureus. But we had to go long way to go, and once it shows very good activity, we have to take it to the next level, toxicology, pharmacokinetics, and HIT has to be developed into lead. So now we are synthesizing, doing the total synthesis of those molecules. And also we are utilizing these MDR active molecules as a cream for topical application. So this is the result of a number of collaboration and communication between a number of people, starting from the tribals and then traditional healers. This was the beginning actually. So MSSRF was the beginning, and they connected us to different type of tribals and traditional healers, where we had regular interaction with them, and the hard work of students. Around 9 students completed PhD in the natural product area from my group, and this is our current group. Now 6 students doing their PhD, and with project students and postdocs. So to end, before ending this talk, I would like to give a doha by Kabir Das. Kabir Das says, "Kasturi kundali basai mrg dunde ban mahi. I say gadi gadi ram hai, duniya dekhe nahi." So that is our situation now. We are having a rich resource, a biodiversity reserve, but we don't know, because the root cause of the crisis is the loss of knowledge base relating to raw drugs or these plants. And this knowledge erosion is happening faster than the resource erosion. We have to come together, in fact. These are all locally available low input technology, which can aid in as useful hints for future technological development. That's over now. So thank you. So we can do research like this. We can work on our passion alone like this and pool, or we can come together like this and do wonders. So let us come together and get together as a dynamic team to utilize our rich bioresource of the country. So because I feel that when I is replaced by we even illness tends into wellness. So I would like to thank the CSIR, which is the organization which give me a lot of freedom to do what I want as passion, along with the required duties as a scientist of this institute. This is the passion which I was taking up, and I have to thank one important person, Dr. Suresh Das, our former director of CSIR-NIIST, for motivating and inspiring me to take up this challenging area. Thank you all.
Thank you, Dr. Radhakrishnan, for a detailed talk on this subject, including the physicochemical characterization of the plant products and related things. Thank you so much for keeping the time also. You exactly completed. I think we can have 1 or 2 questions, sir. Can we have 1 or 2 questions? Is anybody have any questions? This is the rare opportunity you are getting, a person who is very expert in the area.
I think they got frightened with these molecules, but there is no need to get frightened. It is really easy to, anybody can do it.
Yeah.
Radhakrishnan sir.
Yes.
Yeah, please.
It was an excellent presentation. So first of all, on behalf of 100 assembled over here, I would like to extend sincere thanks to you, sir, for your time and presence. So I have just one comment or question. So right now, COVID-19 is a great challenge before the scientific community. And in your opinion, how the array of phytochemicals can help in management of this pandemic?
In fact, already, but the more before the coming to the challenge, the problem in India is we don't have an antiviral screening facility or laboratory in the country. Only when COVID started, we are thinking about the screening. So that is the most important point to make. In fact, we have found several of the molecules which is very active against not only COVID is only one of the virus. There are many other viruses in store. So dengue, chikungunya. I think with RGCB, we are already in talk with RGCB and the new virology institute where we can do the screening, I hope. And I was mentioning about the cyclia peltata molecule, and in the Geridacody molecule, that tetrandrin is mainly found in Geridacody and some other Menispermatia family, Amrud family. And that is already in clinical trial in China. But our problem is we don't know our own treasure, no. So we are more excited when we get a publication. But I think, I don't know, all of us have to come together. I used to tell, I want to do maybe a botany, deeper understanding of botany and also Ayurveda. That will help us in solving so many problems. And the traditional knowledge Ayurveda will give a new, new areas for modern biology, because the Ayurveda is the basis, has already has the knowledge in modern biology, and it will aid in new developments in modern biology. The same thing in modern drug development also. I didn't mention about all these pharmacology results of all those things, because then I will not be able to stop one time. So there are so many other things we can do with all this.
Yeah, there is a question from Dr. Stephen.
I have a question.
Yes.
Please.
First of all, I like to ask a simple question.
Yes, sir.
In Calcutta Press Club, I have asked. That is there is any medicine for any viral disease in modern medicine or allopathy? Is there any medicine for any viral disease in modern medicine or allopathy? And another question that I have asked that is,
Sir, can you repeat the first question? I couldn't.
Is there any medicine for any viral disease in allopathy or the so-called modern medicine for any single disease? Can you say a single disease? Then one more question I have asked. Simple. So suppose if we can solve a problem with a stone, or we can solve the problem with a rifle, why we are thinking only about atom bomb? Why we are thinking only about atom bomb? That is what is happening nowadays, in my opinion, because the mumps can be solved with planjana, and in modern medicine there is no medicine at all. There is no medicine for any viral disease in allopathy. So we have to think about. I am not interested in the terms homeo or Ayurveda. I like to use the term plant medicines only. Plant medicines. So my first question, is there any medicine for?
Of course, sir, there are a number of medicines right now for COVID. So I cannot say which are the medicines used and all, because I am not an expert.
That is viral. No. That is totally false. That is totally false. There is no medicine.
Let me come to the point. As a medicinal chemist, my career is based on this process development and all. So in the last year, we have developed a process for one of the COVID drug, molnupiravir. Okay, and that's already being transferred also. So we have to believe it, no? So it's already shows very good activity. 5 days treatment with that antiviral drug will get rid of COVID. So I don't know any other molecules. There may be many, but I am not an expert in the pharmacies area.
If it is given to the patients, if so, there is no need of vaccination, because within 3-4 days, COVID will not be there. Then no need of vaccination.
But sir, to be frank, let me tell you. So what we need is an integration of all branches.
So that's what I am saying.
So integration, holistic treatment. So I think these WhatsApp Vaidyans and many other videos are destroying this culture, and I think this is not a forum to discuss all those things. But what we need, the need of the hour for the health care of the country and the society, is integration of all branches. There are plus and minus, and we have to accept it. Nobody is better than it, but we need an integration. That is the answer for all these questions.
Actually, I am
Dr. Nair, you want to comment?
one more comment.
Dr. Nair?
No, that is
Just one second. Just one second. I am in
no
Kolkata.
controversial questions, please.
I am in Kolkata. I was on the way to Darjeeling, and Singona is cultivated in entire world. It is now cultivated in Darjeeling only, and it is 99% effective to prevent. Nobody used the bark developed COVID, and the COVID patients. I got COVID only because after 6 months, myself and Ramesh went to Uti and collected, and for 6 months, after 6 months, its quality lost. That is why I got COVID. And I like to say, in Mercy Hospital, they told me, you are having COVID. And
I
that
think we
COVID,
need a different forum.
Yeah, that's
That is-
okay. So I think
is also very much
since time
related
is-
with the phytochemicals.
Dr.
Very much
Stephen.
related with phytochemicals. Thank you, thank you, thank you, thank you, thank you, thank you.
Thank you, sir. Thank you so much.
Dr. Radhakrishnan, thank you. It was an excellent and very informative talk.
This is the bark.
Let me, Dr. Stephen,
Yeah,
please.
yeah.
Okay. It was an excellent talk, and having done this much of working natural product chemistry, how many molecules you have modified to suit its or to make it more efficient for biological uses?
Yes,
Especially
yeah. Once we find a very good in vitro activity, and after getting the mechanistic studies, after doing mechanistic investigation, then so we go for the functionalization of those molecules. So, for example, and most of the flavonoids and some of these molecules, solubility is the problem. And as you know, when we do the isolation from nature, the carbohydrate part is getting cleaved. So we have an active carbohydrate chemistry program going on. So what we do is we reattach the carbohydrate into it and depending on what type of carbohydrate molecule we need, whether it is a glucose, rhamnose, or mannose, or galactose, we reconnect it and see the activity. And also there will be sometimes it may be toxic, so cytotoxic, and we reduce the cytotoxicity through different modifications and also by converting into non-toxic nanoparticles and so on. That is going on, sir. In fact, we can do a lot actually, but for that, whenever I give talk, let us come together. Let's come together because I have
Yes, sir.
MD and with the students with me working on sanitization. So,
but having identified, characterized so many plant molecules,
yes, yes.
how many of them have been shaped into a product which could go straight to the society?
So one of the thing is straight to society is that elephant apples are. So I have elephant apple thing. That molecule is really very good. Oral squamous, oral cancer effective, and this folk remedy actually. It's a folk remedy, and but there also the problem is the solubility. But as a fruit is better, as a herbal. We have to promote that is why we have a herbal formulation facility also. They are parallelly working on using these things.